Hey Em, what are you reading?
Hi Aina, I am reading an excerpt about metabolism from Emma's notebook.
Metabolism? Heard of it before, but what does it mean?
Metabolism is the process performed by living systems to acquire and use free energy to carry out functions, reactions, and pathways to degrade and synthesize biomolecules.
Does it happen in humans' body too?
Of course. Well, to put it simply, metabolism is the process by which your body converts what you eat and drink into energy. During this complex biochemical process, calories in food and beverages are combined with oxygen to release the energy your body needs to function.
So there are pathways going on during metabolism.
Exactly. The pathways may be divided into two, which are catabolism, which is the breakdown of molecules to obtain energy, followed by anabolism, which is the synthesis of all compounds needed by the cell, such as DNA, RNA, and the synthesis of protein. The pathway is called metabolic pathway.
What about metabolic pathways?
Well guys, I haven't really come to the page where the metabolic pathways are explained though. Emma, can you pause your Netflix for a while? We want to know more about metabolic pathways.
Yeah, Emma, tell us. We would take forever to actually start reading the whole thing.
Sure. Metabolic pathway is a step-by-step series of interconnected biochemical reactions that convert a substrate molecule or molecules through a series of metabolic intermediates to yield a final product or products. It is composed of these two opposite pathways, catabolism and anabolism.
I'm confused right now. What is the difference between catabolic and anabolic reactions?
Catabolic reactions break down larger molecules like carbohydrates, lipids, and proteins into smaller parts. This includes the breakdown of ATP, hydrolysis reactions, and oxidation reactions. Anabolic reactions synthesize larger molecules from smaller constituent parts using ATP as the energy sources. These reactions build bones, muscle masses, and new proteins, lipids, and nucleic acids. Examples of these reactions are dehydration synthesis reactions and reduction reactions.
Oh, okay, I get it now.
Do you happen to have any example about the metabolic pathway?
There are many metabolic pathways in every living thing. In humans, the most important metabolic pathways are glycolysis, Krebs cycle, oxidative phosphorylation, pentose phosphate pathway, urea cycle, fatty acid beta oxidation, and gluconeogenesis.
Is that all to it, Emma? Because I've heard enzymes are involved too.
Oh yes! The main key for all metabolic reactions to take place non-spontaneously are called enzymes.
I've heard about enzymes, but what is it about?
It is a globular protein that catalyzes a biological reaction. It is very important for every reaction step to be facilitated or catalyzed. But regarding the metabolic pathways, I still have some problems understanding these three pathways entirely.
Oh yeah? I think Eileen watched a TV series about these pathways. Care to share your summary, Eileen?
Yep, let's talk about glycolysis. Glycolysis is a series of reactions that extract energy from glucose by splitting into two 3-carbon molecules known as pyruvates.
Where does this biological process take place, Eileen?
Glycolysis takes place in the cytosol of a cell, and it can be broken down into two phases.
Wait, let me guess. Energy requiring phase and energy releasing phase, aren't it?
Exactly.
Can you explain more on energy releasing phase?
In energy-requiring phase, the starting material of glucose gets rearranged and two phosphate groups are attached to it. The phosphate group aid in the modification sugar, in which sugar is known as the fructose 1,6-biphosphate. As it is unstable in that form, it will split in half and form two phosphate-bearing 3-carbon sugars. Due to the phosphate used in these steps come from ATP, two ATP molecules get used up.
I see. How about energy releasing phase?
In this phase, each 3-carbon sugar is converted into another 3-carbon molecule, pyruvate, via a series of reactions. In these reactions, two ATP molecules and one NADH molecule are made. This phase takes place twice, one for each of the two 3-carbon sugars. Thus, by the end of the reaction, it will produce 4 ATP and 2 NADH overall.
Oh, by the way, does this reaction require enzyme?
Yep. The most important enzyme for regulations of glycolysis is phosphofructokinase, which catalyzes formation of the unstable 2-phosphate sugar molecule, fructose 1,6-biphosphate.
Ah, I get it now. Based on what you have explained just now, overall glycolysis converts one 6-carbon molecule of glucose into two 3-carbon molecules of pyruvates. Thus, the net products of these processes are two molecules in of ATP and two molecules of NADH.
Yes, exactly. Eventually, there are 4 ATP produced. However, 2 ATP are used up.
Oh. Does anyone know what is the function of the NADH and FADH2 that were produced in the Krebs cycle?
The NADH and FADH2 function as an electron carrier in the electron transport system.
Electron transport system? What is that?
Yes, electron transport system is a set of enzymes which is located on the inner mitochondrial membrane, and it is the main source of energy in the aerobic pathway of energy generation. In the ETS, NADH and FADH2 are used to produce ATP in a structure called cytochrome. Energy released by the electron is captured as ATP by ADP molecule. The ADP then is reduced to ATP by the gains of electron via the process called oxidative phosphorylation.
Wait, what is oxidative phosphorylation again?
Oxidative phosphorylation is the gradient exchange of H+ ion across the inner mitochondrial membrane.
It sounds interesting. Maybe you can explain more about the process, I guess?
Sure. The process in the electron transport system begins when the NADH and FADH2 enter the ETS, yielding 3 ATP per NADH, and FADH2 enter at coenzyme Q, producing only 2 ATP per FADH2. NADH and FADH2 carry protons and electrons to the electron transport chain located in the membrane. Energy from the transfer of electron along the chain transport proton across the membrane and create an electrochemical gradient. As the accumulating proton follow the electrochemical gradient back across the membrane through an ATP synthase complex, the movement of the proton provides energy for synthesizing ATP from ADP and phosphate. At the end of ETS, 2 proton, 2 electron, and half of an oxygen molecule combine to form water. Since oxygen is the final electron acceptor, the process is called aerobic respiration.
Oh, I see. Thank you, Isham.
Since we have heard about glycolysis and electron transport, let's move to one of the most complicated cycle I have ever seen, Krebs cycle. How can Hans Krebs even discover them?
Well. Krebs cycle consists of 7 important steps. The first step is a condensation step, combining the 2-carbon acetyl group from acetyl-CoA with a 4-carbon oxaloacetate molecule to form a 6-carbon molecule of citrate. Second, the product from the first step, which is the citrate, will lose 1 water molecule and gain another as the citrate is converted into its isomer, isocitrate. For the third step, isocitrate is oxidized, producing a 5-carbon molecule alpha-ketoglutarate together with a molecule of carbon dioxide and 2 electrons, which reduce NAD+ to NADH. Thus, in the third and fourth step, both oxidation and decarboxylation occur. They will release electrons that reduce NAD+ to NADH and release carboxyl groups that form carbon dioxide molecules. Wait guys! Can you catch up the first half of the cycle?
There is a lot, Aina, but I think we managed to catch up, right, mates?
Well, no lies in what she says though. Hoping the second half will not be as complicated.
Okay. In the fifth step, a phosphate group is substituted for coenzyme and a high energy bond is formed. This energy is used in substrate level phosphorylation to form either guanine triphosphate (GTP) or adenosine triphosphate (ATP). There are two forms of the enzyme in this step, which is the isoenzymes, depending upon the type of animal tissue in which they are found. One form is found in tissue that use large amounts of ATP, so they produce more ATP. They are mostly found in heart and skeletal muscle. The second form of the enzyme is found in tissue that have a high number of anabolic pathways, so they form GTP. They are mostly found in the liver.
Phew. The important point is phosphorylation to form GTP or ATP, right?
Yeah, that is right. However, dehydration process happens in the sixth step. It converts the succinate into fumarate, which later the two hydrogen atoms are transferred to FAD, producing FADH2. Finally, water is added to the fumarate and malate is produced. The last step in the citric acid cycle regenerates oxaloacetate by oxidizing a malate. Another molecule of NADH is produced. Later, everything will repeat in cycle. So for further understanding, you may refer to our PBR report.
Now, I finally get to fully understand my nose entirely. Ha 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 ha.
Wait, all this time you never grasped the explanation from what you have written?
Jeez, what a klutz she can be.
Hey, don't peer pressure her like that. At least we got to share our facts, don't you think so?
Aw, thank you, Aina.
Haha, thanks.